Today, we have a webinar that we are presenting called An Introduction to Online Dataset Resources for Biological Scientists. I'm John Draper, Program Director for Research and Partnerships at the Stem Cell Network. And I'll give you a little bit of introduction to the webinar, and then I'll be handing over to my colleague, Dr. Megan Mahoney, who's the Director of Scientific Affairs at Training and Programs at BioCanRx, who then will introduce our speakers. So, large biological data sets are essential components of modern biology, including cancer and regenerative medicine. There are now numerous online repositories that house extant data sets that can assist when exploring or validating biological trends. Understanding and evaluating the data sets resources available can accelerate progress and promote research rigor. SCN and BioCanRx are excited to host today's webinar that is designed to introduce researchers to a range of online data resources, including TCGA, GEO, dbGAP, and the GTEx portal that may be used to support biomedical research. Today's webinar, which is hosted by, as I said, Stem Cell Network and BioCanRx, will describe the strengths and limitations of these resources, how to find appropriate data, outline methods for reviewing data set quality, and provide use cases for such an analysis. The webinar will focus on RNASeq data with some discussion of other experimental data types. As always, this is an interactive webinar, so please ask your questions through the Q&A function, which you can see on your screen. We'll save those questions and then put them to the speakers at the end of the talks. So at this point, I'll hand over to you, Megan, and please go ahead and introduce our speakers.
Great, thank you, John. So I'd first like to echo a thanks to our speakers for today's webinar, which really does serve as an introduction. And so with that being said, I'd first like to introduce Samer Hussain, who is an assistant professor and researcher at the University of Laval in the oncology division, and also affiliated with CHU Quebec and the University of Laval Research Center. So Samer has published quite significantly and has published seminal work, in the field of reprogramming, demonstrating several key findings on how reprogramming to iPSCs affects the chromatin state, genetic stability, and gene expression of cells undergoing this process of induced cell fate change. He established his lab in 2016, and his work now focuses on the use of RNA sequencing to investigate the role of long non-coding RNA in reprogramming and pluripotency. So our second speaker today is Gareth, or actually I think he's our first, I'm doing this in reverse order, is Gareth Palladwar, who is a manager of the Ottawa Bioinformatics Core Facility, which supports the University of Ottawa, the Ottawa Hospital Research Institute. He has worked in bioinformatics for the last 18 years, and he has a team at the Ottawa Hospital Research Institute, and it's a core facility. He has extensive expertise in training and education within bioinformatics. He leads an RNAseq workshop that I believe many of you have participated in, that's hosted by SCN that runs on an annual basis. And he also just recently delivered a workshop on the fundamentals of R for both Stem Cell Network and BioCanRx HQP. So with that being said, I think I am first turning it over to Gareth. Is that right? We're on track for that?
Yeah, so that's okay for you.
Perfect. Wonderful. Yeah. So my abbreviation, my introductions were short. So I maybe first welcome any additional information that you'd like to tell the audience.
Sure. I'm just going to, 1 second, I'm just going to spin up my presentation. 1 moment. So I think I can share. This should be visible. Okay, great. I guess I'll just start. So this is a presentation on online high-throughput sequencing data sources and applications. So we're not really doing first speaker, second speaker. We're doing first speaker, second speaker, and first speaker again. So we're going to go back and forth, and Samer and I are going back and forth on the same presentation. So we'll pass off to each other. I'm going to start and introduce some things. Samer's going to present some other things, and then I'll continue with, so he's going to talk about, anyway, we'll discuss that. First and foremost, when you're looking at online data, you have to consider the idea of process versus raw data. In the presentation, we're going to first talk about process data. Samer is going to mostly cover process data and examples of available websites. User-friendly data integration of TCGA websites, we'll talk about that. Author-driven and publication-related websites, and data in publications, I'll talk a little bit about that. My focus later on in the presentation will be largely on raw sequencing data and reprocessing that, how to find it, approaches to using it, evaluating data quality, and at the end, I'll give a series of use cases which might give you ideas about how one could utilize data out there and find it. Online sequencing data sources. This is a very complex thing. I tried to come up with a nice summary of online sources, but there is a very complex and confusing relationship between a bunch of sources that are semi-related. And rather than giving a detailed overview, I decided it was best to focus primarily, we decided it was best to focus primarily on just a few things: the Cancer Genome Project, Genome Expression Omnibus, dbGAP, which is the database of genotypes and phenotypes, sequence read archives, and these are all related, and we'll discuss that. And Samer will also discuss TCGA and its association with various web-based analysis tools, and also web-based analysis tools related to publications, for example. This is not, as I stated, a comprehensive overview. There are many other data sources we're not covering. For example, ENCODE, and one thing I just found out about is China has their own sequencing archive site, which I'll be looking at for data that might be relevant to researchers we support. Okay, so the main data sources I mentioned are TCGA, which is the Cancer Genome Atlas, which is an enormous research project, which is long running, and has over 20,000 cancer samples matched to normal in many cases, spanning 33 cancer types. It's a fantastic resource, accessible via the Genomic Data Commons data portal. Once again, we'll talk about that. Gene Expression Omnibus contains a lot of process data relevant to particular publications. In general, publications which have sequencing data are expected to deposit that data in Geo and through that to SRA, the Sequence Read Archives. And that's kind of a hook into where you can get the data associated with a given publication. The Sequence Read Archives contains the raw sequencing data and is referenced by Geo records and other sources. So that's where you get it, the actual raw stuff that came off the sequencer, which is then processed into the analyzed data. And dbGAP, which is the database of genotypes and phenotypes, contains human study data, most of which is controlled access. So this is where we get access to things that are maybe not like directly accessible and always accessible. Sorry, not directly accessible without permission, and we'll discuss that in permission. So Samer is going to take over now, talk about TCGA and various sources of analysis tools online.
Okay, first, before I start, I would like to thank Megan and John and also Gareth for the nice introduction. Megan, I don't have much to add really. Thank you so much for the introduction. And like Gareth said, I'll be focusing a lot on website-based data. So the majority is on TCGA and cancer related, but I added a few things because I know a lot of the people here might not be working on cancer. And so I added a few things based on some of my interests, which is neural development and prepotency or early development. So just to start, I just want to give you a brief history on TCGA, the Cancer Genome Atlas. And it's really started, like just briefly, it started in 2005 as a pilot plan, and then it really started to, in recent years, started to really blow up. People started adding more data to it, and it's not just TCGA related, but any cancer data that, and I'll show you later some examples. I mean, you can see on the right here the amount of data that's being compiled in the TCGA. I mean, it's just massive. And anybody that's working in cancer should really, one of the first things we should do, we should always look at these data sets. If you want to know more about the history, then you can go to this website here. So there's two websites. There might be others, but the two websites that I really use, one is very well, CBioPortal is very well maintained. It includes both TCGA and non-TCGA data sets that are manually curated, and you have more than 200 studies with more than 60,000 samples within this cohort of data. It's maintained by multiple institutions like the Memorial Sloan Kettering Cancer Center, Dana-Farber, and also our dear friends in Princess Margaret Cancer Center in Toronto. So I mean, there's a whole. It's a nice website, and I'll show you some of the features of this website. But it's a really nice website to get started. You just have to be careful when you use any of, and this one I want to emphasize, and I think Gareth will emphasize it later, but when you use any of those websites, you just have to be careful what you're looking at. I mean, and how the data is analyzed. This should serve more of a way to get to the data analysis rather than your final analysis per se. Gareth, correct me if I'm wrong also, if I say anything that contradicts that. So CBioPortal, you have, as I mentioned, you can look at multiple types of cancers. You have different data types, but the majority is RNA-seq, microarrays, or whole genome sequencing, and it has to do a lot of mutation studies or expression studies. You can, if you want, just follow the publications and look and see, okay, if there's other types of data. Like, for example, some publications will have genome data or chip sequencing data that is integrated with the RNA-seq, but on the CBioPortal is mainly RNA sequencing or whole genome sequencing. What's really nice is you have a lot of patient samples that you might not be able to have access to. So, and with clinical information like whether the patients had drug resistance, survival of the patients, also like within how the gene amplifications or how much of the genome was really altered within these patient samples. I'll use it as an example today, brain tumors. So here, if you just check into the brain tumor section of these data sets, you'll have multiple. You have different types of brain tumors, for example, and you can have from the same study, you can have multiple different types of data on different brain tumors, or you can collect them from separate studies. So again, when you start to do from separate studies, you have to be careful because they're not analyzed always the same way. So you just have that in mind when you're looking at this data. And here they highlighted also some studies have overlapping samples. So they took data from other samples from different studies. You just keep that in mind. So once you select, let's say, a type of cancer you're looking into, you can explore in two ways, whether you go gene specific or you can just take all of the studies you've selected and just explore that. And here, the next step is, okay, once you select this, then you'll have all the list of, for example, like I said, I'm looking only specifically at gliomas. I have different compartments within the website where you will have, for example, the cancer types that you want to look at. If you want to compare, let's say, low grade versus high grade brain tumors, then you can select the data sets that have that. You can look at gene amplifications. You can see like the top genes, which ones are amplified the highest in most of these. Again, you have to be careful if this is coming from one study or is coming from multiple studies. So by clicking, you can see, let's say here, you can see which studies it's coming from. For example, here you have IDH1, which is highly mutated in gliomas, but that's specifically in, I'll show you later, in low-grade gliomas rather than GBMs. But you see that it shows here that it's highly mutated in these data sets. But that's coming from really one or two studies rather than all of them. So you really have to be careful when you're using it to make sure that you're not making false conclusions. So you have different types of analyses. You can look at copy number changes, RNA expression, mutation load, like here, we show it here. You can also compare different types of tumors. Like, for example, I wanted to compare, for example, diffused glioma, which is specific to a certain region of the brain, versus glioblastoma multiforme. And then from that, you have the option of comparing these types of cancers and looking at the survival rate of the patients from these two different samples, or the clinical aspects of these samples. So one thing here is not apparent, but sometimes from the same patient, you have multiple samples. So the number of samples might differ from the number of patients that you see. So that's also important. So you can always go back and check, and I'll show you later which patients have, let's say, multiple samples and what those samples are. Like, for example, it's post-treatment or pre-treatment type of samples. So that's the thing. It's really, to some, it is user-friendly. The other thing is well-maintained. And if you have any questions, you can always, they're very reactive. You can always go back and send them an email if you don't understand something, or if you need some clarification or some type of analysis that they may or may not offer. The other, like I said, you can explore by the selected studies, or you can just input either one gene or multiple genes together if you have a gene signature, for example, that you're interested to see. Here I picked IDH1 because, like we saw earlier, it's really one of the highest mutated in low-grade tumors. So I'll show you now. And here in this one, and you can also select if you want if you put in, let's say, if you search for IDH1, you can also select if you want to include or not data on mutations or copy number alterations. And please feel free to post your questions if you don't understand any terminology that I've been using in this presentation. Like I said, so when we look at ADH1, let's say I looked at cancer type summary. So it lists all the summaries and studies that have this mutations in this. For example, here, the UCSF glioma studies or sample, this IDH1 was mutated 100% of the samples. And then you see in the low-grade glioma, you have less, like more, for example, than the GBM, the glioblastoma multiforme. So, and also you have to go back and see what are those samples, where are the samples and what they mean for the patient, for example, and survival rates and so on. The other option you have is, if you don't want to go through selecting, you can just type in and search. Let's say here again, I just use glioma as an example, but you can put prostate cancer or any other types of or breast cancer or any other types of cancer data sets. And I can see if I type in glioma, I have multiple data sets, and this list goes on. I didn't show the whole thing, but you can also see the reference for which paper it came from. This, for example, this came from Memorial Sloan Kettering study, or you have the TCGA study, which involves multiple sites as well, and different types of publications. So you can go by publication base and just specifically, let's say, look at low-grade gliomas, or you can go deeper and look at all of them together. You have that option as well. You can download some of the data. Some of them are, I think Gareth will show you what is access controlled and what is not. But some of them you can just download the data in text format, or they call it tab delimited format, and you can open this really in Excel and look at it, and you have a matrix which tells you what samples goes with what expression data. So for this, you might want to ask someone that's familiar with bioinformatics a little bit, because if you just download it and you don't understand the numbers, it might not be very beneficial for you. But if you have someone near you that understands, let's say, Excel or bioinformatics a little bit, downloading this data will help. They could help you really, okay, this is what this means, this is what that means, and being able to put it together for you can actually just look at it in Excel or something like that. You also have sometimes expression data that's already analyzed, like looking at Z-scores instead of the raw counts data, or an expression like FPKM or RPKM, so you don't have to normalize anything. You have those options if you want to download the data. So that's really more for CBioPortal that I'll just cover. The other thing I want to cover is, it's a website I recently found, I'll say 2 or 3 years ago. It's called GPA. And this website compares TCGA data to GTEx, GTEx, which is genome tissue expression data from that was collected from NCB, and you'll find that mostly in NCBI. And if you're really interested, you can go on the GTEx portal, and if you want to look at your, let's say, a specific gene or specific gene sets, you can go to this website here, GTEx portal. You can see what tissues is your gene, and it's mostly human data. What tissues is in what tissues are your gene or your gene sets are expressed in. Again, I have to emphasize that you have to be careful when you're using these kinds of websites, especially if you don't see them in multiple publications. CBioPortal, I'll say, is very reliable. This one, I have to admit, I have not tested it enough to say that, okay, it's very reliable or not. So that's why I would suggest if you use it, you always look, be careful on what the conclusions you have, what conclusions comes out of it. What is nice is with these kind of websites is it gives you an idea, okay, this is my gene. I want to see how it is in different types of cancers have been involved and compared to normal tissue. Again, they're comparing tumor cells to normal tissue samples, so it might not be the best comparison sometimes because of the nature of the cancer. And the mix of cells, let's say, in the tissue versus the mix of cells of the cancer. So it might not be the best comparison. It just gives an idea. So that's why be careful when you're using this data set. So essentially, you have this kind of home page where you can just type in your gene here. And for example, I tried again IDH1, and then what you can get is the expression patterns where IDH1 is expressed, but you have different options. You can have isoform expression of IDH1, you can have survival analysis, and I'll show that later, differential gene expression, or also correlation, for example, with other gene sets. This is something that I find useful to see if patches, like for example, I looked at one point at some of our prepotency genes to see if they are correlating with other prepotency genes in these data sets. So it kind of gives you a validation of how the analysis from this comes out. So this is what comes out when initially, so you can compare normal versus tumor tissue, like I said, and here it's a nice representation of here how the gene is expressed in the different tissues versus normal tissues. So here, like IDH1 is highly expressed in, for example, brain tumors versus normal brain tissue. And they have a different way, other ways to represent. So essentially here, if you have, again, like it's in GBMs, which is highlighted here in red, it's more significant, the expression of IDH1. Same thing for low-grade glioma. So you have the definition for these within their website as well, so you can look it up, what the definitions of these different types of cancers. But you can see, so at least it tells you in what cancers it's expressed more than the normal tissue within that. But again, you have to be careful if you're going to use this, that you don't draw definitive conclusions. This gene is really more expressed in cancer cells versus neurons, or type of thing, type of conclusion. So I think because you have a mix, when you look at tissue samples, you have a mix of cells, so it's not necessarily a certain type of cell. And I'll get into websites, some websites that you can, if you're really interested in neural development or things like this, I'm going to get into it later on. So the other way also you can look is you can type in your gene, like when I said like survival assays or survival analysis on different patients, how this gene, or you can also put in a list of genes. Now you can get different an analysis of if this gene is expressed, what is the prognosis of certain patients. So that's the nice thing here. It focuses mainly on the tumor or TCGA data. And you can select the multiple data sets. If you have here, I selected low-grade glioma and glioblastoma multiforme. And essentially the data that comes out of this after is your survival curves. So if it is highly expressed in the different patients, you have a lower survival rate than lowly expressed, as well as the similar kind of curve. And also it calculates the significance for disease-free survival. So this is really the end of the CBioPortal and TC and GPA websites. I'm gonna get into more now, like publication specific websites, and this is just 4 that it goes back to what my interests are. And if you ever need to find data, for example, that is for the subject that you're interested in, we can work together to see if there's other publications that show it. So these are very publication based, and it's a very nice thing from these groups because they not only share the data, but they present a website that helps you go through their data, and there's a lot of explanations. I'm going to show each one of those and how it works. For example, one of the first ones that I really liked was they called it Corticon dataset. It's basically RNA sequencing data on human ES cell differentiation towards neurons, and what they did was they picked at different time points. So it was from Dr. Sally Temple's lab in the Neural Subset Institute in New York. This is one of the oldest ones I've ever worked with. It's in 2014. And what's nice is basically you can download the data and the scripts if you want to reduce their stuff. But really, and I highlighted it here, but you have to really pay attention because if you were to look for a gene and you see it's not behaving the same way here, you have to just keep in mind as a let's say a stem cell biologist that not all differentiation protocols are the same, and this is based on a more older version of the neural differentiation protocol. So maybe you're using a different protocol and you have more specified cells. So this is really a general differentiation protocol that. So it's just to give you an idea where your gene stands in the differentiation process. And what they did, it was really nice, was they separated into different clusters. You have the cells prepotency, the ES cells or ESL cluster. You have the early markers of neural differentiation cluster, which you just see a little bit of expression in the early days. You have cortical specification. Also, you have what they call or deep layers or upper layers of the cortex to see. The other thing is you can search by gene. You can just type in whatever gene you like. You can browse. The one thing that I added is the disease association, and I'll show it a little bit after. You can look by pathways. If you're not familiar with tech pathways, these are just signaling pathways, a list of signaling pathways that have been curated, and a lot of people work with them to find out what the pathways that are involved in when you analyze your data. You have also the GO-term ontology, like biological processes, for example, development, neural differentiation, sometimes some signaling pathways as well in this. And what was interesting in this website also is they included multiple disease associations, not just neural. For example, I looked for Parkin, which is a gene involved in Parkinson's, and there's multiple things for here. For example, it might be associated with renal carcinoma. So the thing with these associations, again, you have to be careful when you see this kind of data, and make sure that it could be validated by other types of analyses, not just this type of analysis. The other thing here, it tells you that Parkin is expressed mainly during the early neural differentiation and the cortical specification, and then it goes down. So you can see the expression data here, it goes up during the first days of neural differentiation, and then it starts to go down a little bit. So you can do this for multiple genes as well if you want. Next, another data set which I found recently. It looks at mouse gastrulation, single cell RNA-seq. This is very, very useful if you work in early development. You can see some of your genes, how they behave, how they are expressed during early development. For example, I just took nanog as an example, well-known prepotency genes. This is the homepage of the website. You can click on gene interrogation tab here or different things, but all of this stuff you can always go back paper what this means, or follow the website, really go step by step. And I'll show you can also download some of this data, some of the visualization of this data using these options here, and or you can just visualize them using the gene interrogation. option. So this is how some of the single cell UMAP visualizations that you can download from this. You can choose the time point that you want. Sorry, I'll go back just a little bit. You can choose the time point that goes up to E12.5, I believe. You can choose the cell type if you just want to go by cell type. There's also the different types of visualization like UMAP or and essentially here I chose UMAP, for example. And I can see that the expression of nanog is very limited at E7.0, limited to primitive streak and to some extent the epiblast, which makes sense. But again, you can look at your gene of interest to see where it's expressed. The other data sets, like another example where I find Sten Linnarsson's lab in Sweden, they have multiple data sets on single cell RNA-seq, RNA sequencing, data from mouse and human. Here, for example, the example I'm using here is on ventral midbrain tissue. So they have different examples of brain development, and what's nice about their lab is they not just provide the data, but they also give you all the information you need to be able to reanalyze the same way they did or reproduce their main figures. So this is the publication and the data that I'm going to go through. But if you go on their website, you have the different publications, the data they have, the tools that they have made to analyze and visualize the data. So I find that very, very helpful and really nice from them that they provided. And it's quite useful if you just want to check quickly where your gene is expressed and what cell types it is. For example, here I just typed in NeuroD and I chose NeuroD1. You have mouse and human and mouse data. And basically here, what it's telling me in the ventral midbrain, the NeuroD1 is expressed mostly in neuronal progenitors or medial neuroblasts, which are precursors for dopaminergic neurons. So again, you just type in your gene of interest and you find it. So that's the nice thing about using a website like this is you don't have to go and reanalyze and look at Excel files and all this type of data if you're really interested specifically in something like this. The last one I would like to show, which it's more of an algorithm to predict cell fate conversion. So people in reprogramming might be very interested in this. If you want to order cell programming, and it uses a consortium, the Phantom 5 consortium expression data sets, which is filled with a lot of expression data from different human cell types and tissue samples. So it takes all of that data and basically tries to figure out what transcription factors you need to convert from one cell type to the other. And there's hundreds of conversion possibilities. Like here, I just chose fibroblast to human neuronal samples, and that could be anything really. You can choose like the normal we can do when we do IPS or induced pluripotent stem cell reprogramming. You can just choose fibroblast and type in induced pluripotent stem cells here and you'll get it. You have to basically, it has to be on the list, otherwise it wouldn't work. But the nice thing is it's a very solid, I would say, predictive algorithm, and they have validated some of the stuff in their paper, in the Nature Genetics paper. And what is important is it gives you a list of factors. You can add them all together, but really what they say, to get 95% conversion, for example, you only need these three to get it from fibroblast to human neuronal samples. And what's nice is they give you how the network of how these transcription factors are related. Now, again, this is just a prediction, and yeah, it might give you really a direction, but you might have to play with these factors, let's say, if you want to try it out. And so just all these websites that I've given, maybe just go and try them out quickly. And that's pretty much it for me for this, but if you have questions later, I'll be very happy to answer. And Gareth, I'll let you take over.
All right, so I should be sharing my PowerPoint now.
Yeah.
So Samaras showed us a lot of tools that are web tools for interacting with data that's been integrated in various ways. So those are great, and I've used many of them myself. But one thing to keep in mind is those data sets, those sources are in fact they can be a little ephemeral. You know, it can be the site might go away and the content might change. So I often recommend that people use those sites, for example, for generating hypotheses they test by other means. But reproducibility can be an issue. So it's just something to keep in mind, that you might come back a year later and everything's different. So just something to keep in mind. And this gets into why I like raw data, because I can own the whole analysis from end to end. That's the bioinformaticist in me. So once again, we're talking about TCGA, GEO, SRA, and dbGAP. So I'm going to walk through an example which kind of integrates those all to give you a sense of their relationship and their relationship to raw data. Okay, so GEO is Gene Expression Omnibus. So as I stated, usually the records, or what are called the series, the core records or series in Gene Expression Omnibus, are publication related. So this is just today's Gene Expression Omnibus site, looking at the number of, for example, series expression profiling by high throughput sequencing. There's 55,000 series. Essentially, those are papers. So that's a lot of data sets. And each of those might contain anywhere from 1 to several hundred high throughput sequencing data sets. In GEO, so this is an example of a GEO record that we deposited on behalf of the Rudnicki lab related to a publication. You can see that this is a series. It's related to the publication, which is cited below here. The summary simply describes, it's a copy of the abstract describing the function of the publication. And then if we scroll down, we see there are samples associated with this. So each series has multiple samples. You can in fact have super series. So if you have a publication which has, for example, an RNA-seq data set and an ATAC-seq data set, those can be sub-series of a super series, which is the paper, and then you have a sub-series for each data type, and then samples below that. So in this case, we have 6 samples, 3 replicates of PAX7 wild type, and 3 replicates of replicates of PAX7. I believe they're knockouts. So that's those are the records associated with that. And we see down below, there's an SRA run selector. So that actually is a link to the raw data associated with these series. That, excuse me. So the sample up here, if we look at the first sample, GSM 5106790, which is associated with this specific RNA-seq data set, we see that it has more information specific to, and I'm not going to go through in great detail, the protocol that was used, the organism, various characteristics, and exactly what was done. Essentially, it's the materials and methods for this data. And the specific SRA, the sequences associated with that. Now, GEO doesn't contain, as stated previously, GEO doesn't contain the raw sequencing data. It contains process data or analyzed data in various ways. For example, in this case, there's some quantification data. You might have fold change associated with it. But if we then if we were to click on the series, the series run selector here to see all of the runs, the RNA-seq runs associated with the data, we would see this in SRA. So these are the actual raw RNA-seq files. Now, generally, what happens when you have a, I'm not sure what that false public is. I don't know where that came from. Some sort of error. Anyway, so these are the records associated with these 6 samples that were run. They were submitted as FASTQ files, but in SRA they're converted to an interchange format called .sra, which can be converted into various things. But so this is the relationship between the various elements. Now we talk about SRA, so we have GEO series containing samples. The samples have information in them about the samples, and they refer to an SRA record which contains the raw data. So the SRA database is the sequence read archives containing raw data. Note that this is a graph of the growth of the sequence read archives, and it's grown very quickly over the last few years. It's growing faster and faster all the time, I think, or yeah, moderately fast anyway. But note that we have on the order of, that's a quadrillion, there's a lot of commas there, but that's quadrillion bases of sequence data in there. The key thing to note here is not just the rapid growth, but the fact that 24 quadrillion of those bases are open access, but there are a total of 55 quadrillion bases. Now, some of those bases that haven't been released yet, some of those sequences, I mean, essentially just counting the number of total bases in the sequencing that's been done. But a good chunk of this stuff is restricted access, which means that you can't just access it without permission. So permission is usually restricted because it's patient data, and the patient data is potentially identifying, or it may have consenting restrictions on it based upon what the patient consented to. So that is access, that is controlled through dbGAP, which is a kind of clearinghouse for access to consented and patient data that they want to control in some way. And it's quite a reasonable source. Once I found this, I was able to find all manner of raw data that was particularly useful. In particular, Cancer Genome Atlas, the underlying sequences, if you're doing whole genome sequencing on a cancer or a normal sample from a patient, we now know that with the amount of information out there, that's potentially identifying information. You can figure out who that person is in the long term. So they want to restrict that and control it, which is quite reasonable. So you can search dbGAP for data sources, and there's all manner of data in there. I'll be giving a couple of examples using dbGAP restricted data later. So I strongly recommend, if you're looking at human disease models, or anything in humans, you have a look at the dbGAP data source. You can go to dbGAP and search it, and you may find data sets which are of interest to your research, but you need to generally apply for access to them if you want to use them. As I stated, it's a reasonable request given the nature of the data. It's pretty simple. You have to be a principal investigator. You may require institutional approval. You have to provide institutional contact information. Signing officers have to sign off on it. IT director has to sign off on it to basically say you can actually secure the data. You have to provide a research use statement describing your project, saying what you're going to use it for and agreeing to only use it for that. And then you need to agree to not use it for anything else, and delete the data when you're done. There's a yearly review where you update what you've done with it and or close out the request. It can be a little bit laborious getting the first application done, but once you've done that and your institution is familiar with it, it can be a lot quicker, and it gives you access to data which is not as used as the stuff that's in the open access sites like Geo in general, and makes maybe a little bit, it's not been as raked over and analyzed as the other stuff has. So the Genomic Data Commons portal, this is one of the ways to get at the TCGA data that Samer was discussing from the CBioPortal and the other tools. This is the raw data for it that we're looking at. So you can search here. It gives you the number of sites, files, genes, et cetera, et cetera. So, but this gives you access to raw data, raw-ish data, which has been still identifying, and you can download it. So TCGA, it's a bit confusing, all the elements here. So consider TCGA dbGAP access example, where you're interested in TCGA ovarian cancer whole exome sequencing data, for example. The raw sequencing reader are stored in SRA, which is sequence read archives. So you can download those various tools if you have permission via dbGAP. The variant calls and the map reads, for example, are stored in the Genomic Data Commons portal, and you can download them directly or from other tools. And once again, you can access them if you have permissions that are associated with your login. So reprocessing data and quality assessment. The raw data is great. So data that's out there is great. The publications and Geo contain results usable for reanalysis. So they have stuff that's already been analyzed, and you can have a look at it, like gene expression fold change tables, ChIP-seq peak location, variant calls. And you can work with it as is, and we frequently do, but you have to ask yourself, can you trust the data quality? Can you trust the analysis that they use? Does it make sense? And can you integrate it with data from other sources? Often when you're dealing with imagine you have multiple ChIP-seq or let's say RNA-seq data sets you want to look at, you want to integrate and compare from various papers, you can't really compare data sets and samples that have been processed by different pipelines. They really need to reanalyze them by the same pipeline. So if you want to reanalyze them by your own method, the first thing to do is to assess the quality. The encode, so of course there's all different types of next generation sequencing data. I tend to focus on ChIP-seq and RNA-seq. That's ATAC-seq. That's stuff we've most used ourselves. But the ENCODE consortium is a good source of data standards for quality assessments. So, for example, if you go to the and I've got the link here. If you go to the ENCODE data standards site, they'll have information on what you expect to see for reasonable quality bulk RNA-seq data, for example, total RNA-seq. And it will give you ideas of the depths that you should look at, the duplication rates, the number of replicates, all those things. And you can then evaluate that data that you've downloaded based upon that. One of the things we often use is a tool called FastQC. When we downloaded new raw data, we'll just do a basic analysis of the raw data using FastQC to evaluate, the basic statistics per sequence quality. This is the average quality along the length of a read. You can see that this is an example of a bad sample where the read quality drops over the length of the read rather rapidly. Per sequence quality scores, per base sequence content, GC content, all these things are indicative of, bioinformaticists can look at that and tell you if that makes sense given the data you have. And it won't necessarily tell you if the data is good, but it can indicate if the data is bad. For example, and then it's difficult to talk about this stuff in terms of what should be done. This is an example of potential paths for an RNA-seq analysis pipeline. So, for example, our group tends to use, we trim, and then we use salmon for pseudo-alignment with the raw reads with the RNA-seq, and then we take the pseudo counts from salmon and we compare it with DE-seq2, right? So we have this pipeline of tools we use. Other groups may use completely different pipelines. So in general, I can't say exactly how you should analyze your data, but there are various options available. However, if you get a bioinformatics group or a core facility to analyze your data, one thing you could ask them to do is integrate the pipeline results, because there's usually multiple steps in a pipeline that various tools run. Integrate those results with multi-QC, which will integrate those all in a nice, pretty report. It's a lovely tool. We use it all the time to have a look at many samples when they've been analyzed. I've used it for hundreds of samples at a time, and it works really well to get an overview of what's going on. Then you can review the quality of the data, look at the number of reads, for example. In this case, we have general statistics, and it's saying for this sample, which is actually a SRA sample, the number of assigned reads, the percent assigned reads, the number of assigned reads, the number of trimmed base pairs, the number of duplicates, etc. So it gives you a sense of the quality of the sample. So if you're starting to analyze with raw data, and I think I'm going to try and finish off with just a couple of examples. You want to start with a hypothesis. You want to curate a set of publications that contain data of interest that could test or falsify that hypothesis. You want to work with bioinformaticists to create a data analysis plan if you can. And then for very large data sets, if you've decided to proceed with the analysis, take a small subset of that data to validate the pipeline and output. Once that's given the output you want, so you've determined an end-to-end, it's giving you the output a smaller subset of output from a smaller subset of data that makes sense, and then you can run the whole thing. One thing to keep in mind is there's economies of scale here. Most of the work is setting up the pipeline, so 20 samples analyzed won't be, that should be 200 samples analyzed won't be 20 times the price of 10 samples. So there's a lot of benefit in going big if you want to. And then you can also incrementally add data as you find it once you have the pipeline in place. So I'm going to give, I've got a bunch of examples here, but we're kind of running out of time. So I'm just going to give 2 examples. This is an example where I took raw data, processed data from publication for Dr. Tamara Ogilvy's lab at University of Manitoba. She was doing some medulloblastoma. This is from a paper on medulloblastoma. And she was looking at expression of OTX2, PAX3, and PAX6. And from her samples, she had some histone marks, ChIP-seq data of histones, and the some RNA-seq data. So to complement that, she found some data in a paper by Boulay et al., which is GeoSeries GSE 92585, which had been mapped to HD19. I remapped that to HD38 and made a nice visualization of it around these genes to show the effect of those histones, the chromatin modifications in those areas. So I guess the other one that I'd like to pick, I've got a bunch, but I don't really have time to go through them all. This is a really large-scale analysis that I've done for Dr. Baltz at the Ottawa Hospital Research Institute. Dr. Baltz studies oocyte maturation and is looking at multiple species. So with the help of his grad student, Angus Macaulay, Angus curated a huge set of papers and the associated samples, which we then integrated into 3 separate data sets of RNA-seq data. And the RNA-seq data was integrated and analyzed as such. And this gave him a good view into oocyte maturation across species, as not a tool for publication necessarily, but something that they could use in their lab to kind of test hypothesis. Is this gene expressed under these circumstances? The important takeaway of this is that we have multiple samples across multiple experiments, and they integrate pretty well. And you can see the time series, which goes from GM to 1-cell, 2-cell, 4-cell, 8-cell morulloblast. Actually, in this PCA plot shows a nice smooth transition. And these were a trajectory. And these samples are, in fact, from many different experiments. So at this point, no data has like batch correction or anything has been done, and it's still usable data. So I think I'm at 5 minutes, 2:55. So I'm going to finish up here, and I can go through some other examples if people want to see them later, or I can contact me about them. Just finishing up my presentation, and I think Samer as well. I'd like to acknowledge Stem Cell Network, the University of Ottawa. I think Samer was acknowledging University of Laval and CHU at Quebec and TCL, the Ottawa Hospital Research Institute, and then the various use cases I didn't fully present are from Jay Baltz, Dr. William Stanford's lab, Dr. Derek Gibbing's lab, Dr. Tamara Ogilvy's lab, and these were all examples of analysis that were driven by the scientist requests. Thank you very much.
Fantastic. Thank you very much, both of you, Samer and Gareth. At this point, please do enter questions into the Q&A function. And I'll start with a question to you, Gareth. The papers that you see published in your field, how important is it to go back and actually look at those data in terms of the quality that the quality of the data and the analysis that they've used, and try and reproduce it when you're starting out, for instance, on a project that's going to be focused in that area?
I feel, yeah, my mic's on. I feel it's important it depends what your area of interest is. If, for example, you're starting out in an area researching a topic, the idea is not a common one, but I think one that should be promoted is of a data review, just like you would do a literature review. Someone can imagine pulling together all the RNA-seq data for a given paper series or the thing you're studying, and then looking at it directly, checking the quality, verifying things work. I mean, in practice, it's not always practical. The data is generally very good, and it's not always practical or quick to verify results. But if that is your specific research topic, and you question a result, then going back and reviewing exactly what they did is worthwhile to determine if it makes sense. And also, different pipelines can give different results. So the other question becomes, even if the data quality is good by those criteria I stated, does a slightly different pipeline give a similar result that makes sense? How robust is that result, or is it very sensitive to the specific pipeline that we're using?
And it's important to be saving those pipelines, and actually, because they're essentially the methods for how you've analyzed the data, aren't they? So,
yes.
Samer, I've got a question for you here. So, what are the common mistakes that you see trainees making when they're starting to analyze data for the first time?
Oh, when we're talking about the websites or raw data?
Either.
Either, okay. I think it's just one of the things that, for example, even with raw data, Gareth, maybe you can help me out with that as well. But what I find with maybe with trainees is they don't consider all the, let's say, controls, like the different ways of analyzing that can show the same point or not. So just trying to think of an example. Let's say if you're seeing a bias for a certain condition, is that bias due because, let's say, example, let's say you're seeing a bias towards protein coding genes versus long coding RNAs. Is that bias, for example, because you have, in general, you have more protein coding genes in the genome versus long coding RNAs? So that has to be taken into consideration. So if they don't look at, for example, the frequency of protein coding genes versus long coding RNAs in the genome, and they just look at their data, just, okay, I have more proteins being affected versus long coding RNAs, then the conclusion that they have is not real, because they didn't really take the control of looking, okay, do we have more protein coding genes versus long coding RNAs in the genome? And is that why I see that difference?
Yeah, that issue of choosing the wrong background can be a serious one. Where you can get a lot of you can get a lot of bias as a result of that. Yeah.
So that's one thing I've seen. Like in terms of the websites, it's I tried. I don't know if I emphasized it well enough, but if you're using the websites, you have to make sure what you're looking at. Like for example, GPA, I use it, or I would use it mainly as a first look. Okay, is there something interesting? And then, like Gareth said, I will just go back and see if I can find it in a different data set or a different publication or format. If it's not in a publication, then you have to dig biologically, maybe try to see if I address that question biologically. Because you're comparing sometimes apples and oranges in those analyses sometimes.
We only have a minute left, and I just want to ask, for people who are starting out and they're kind of struggling with their analysis, what resources or experts should they turn to at that point to make sure that they're not going off in the wrong direction?
I mean, being in the bioinformatics core facility or a bioinformatics core facility, I recommend that anyone contact their local bioinformatics core. In general, they will discuss with you and they'll give you a sense of how to approach things. I've encountered a lot of students who've spent a long time trying to do things on their own without kind of just discussing approaches with a more skilled bioinformatician, or let's say more experienced bioinformaticist. And there's a lot of things that can be problems that can be avoided in basic standards that can be applied that students starting out may not be familiar with. So yeah, contact your local experts and have a chat with them.
Okay, well, fantastic. Sorry, go ahead.
I was just going to say, re-emphasize something that maybe focus, if they want to learn, focus on how rather than just spending a lot of time doing things that are unnecessary. Because a lot of times it could be done in 1 hour, what would take them, let's say, 10 days. So going back to a bioinformatician, like Gareth said, would be the best option.
Fantastic. So some great content there, and thank you very much to both of you for presenting, Gareth and Sama. Very much appreciate it.
Thanks.
And thank you to all of our attendees for tuning in. Okay, see you next time.
Very much.
Thank you very much. Yeah. See you.
Bye.